They brought me in this year to give you kind of a very brief overview of biology. I'm not going to teach you all of biology, but I am going to give you some familiarity. In this packet that I made for you, you actually may have teachers who use worksheets similar to this. You may have actually done these worksheets before, which is fine, but I kind of, as much as I could, try to put together everything that seems like this is the base level you would need for biology. All right, so let's get started. So we're getting to my favorite part, which is gene expression, how we make proteins, because it's the central dogma, right? It's the most important thing. This idea that DNA makes proteins, they call that the central dogma, because all living things either have DNA or RNA to make proteins. If you're in a lab, they'll say, don't you know the central dogma? And in high school, I didn't know the central dogma. But the central dogma is basically just DNA transcribes to
RNA.
RNA, very good. And that makes proteins, yeah. And we'll actually talk a little bit more specifically about this. But everything that you are is proteins. They say like DNA is the most important thing. It is, but only because it makes proteins. DNA doesn't do anything, right? It's just some instructions. The things that make the proteins are there. So this is why even the most basic cells often have to have ribosome.
What RNA stand for?
Oh, RNA stands for, I'll actually write this down. We'll learn this a little more. It's ribonucleic acid. And DNA is?
Deoxyribonucleic.
Yeah, deoxyribonucleic acid. But many of you are going to work in a probably genetics laboratory. It's helpful to know the difference between those two. Okay? So in terms of living things, you either have to have DNA or RNA, some genetic material. But there's something that doesn't necessarily have DNA or RNA, but it's just almost not alive. Yeah, viruses. So the one kind of exception there is like viruses. And a lot of people say the viruses aren't living. I don't know why they say they aren't living. Yeah?
They're unable to manufacture their own proteins or viruses so that they can sustain themselves.
Yeah, they can't do the central dogma, right? They can't make their own proteins. But what they do is they hijack cells, right? They go into cells and they get cells to make things for them. And we'll actually talk about that later on. But viruses are kind of like semi-alive. You're all definitely alive. You all have DNA that makes these proteins, right? Your whole body's made of proteins. Your eye color is the eye color it is because of the shape of the proteins. Your skin color is the color it is because of the shape of the proteins. But viruses are really cool. And if you ever work in a virus lab, there's a lot of different ways that viruses take advantage of things, okay? So we're gonna jump to gene expression. And you gotta trust me that I'm sure a lot of you have a basic understanding of this. And it's the kind of thing that it seems complicated, but if you understand these diagrams, this actually becomes a pretty eloquent system. And eventually what we're gonna get to is we're gonna get to a place where we understand the difference between an intron and an exon. You might have heard of these called junk DNA. They're not necessarily junk DNA. But we're gonna try to figure out what of our DNA kind of makes proteins and which of it doesn't. Why doesn't it make proteins? All right, so let's get started with this. All right, so I'm going to have us jump to, we're going to skip the Y because we've already read quite a bit about that. Let's go to page 2. All right, consider the eukaryotic cell in Model 1. Are you considering it? Okay. So for 1A, where in the cell is the DNA found? You know the answer to this. It is in the nucleus. For 1B, where in the cell does transcription take place? Well, this is transcription, and this is all in the nucleus. Good, so transcription takes place in the nucleus. Number 2, what polymer is like a long strand of something, or a long chain of something, is being synthesizes to make something during transcription? I am growing RNA, but specifically I'm growing something called pre-mRNA. And I want to make sure that you get that down because there's a little bit of a difference. And if you were working in a genetics lab, the difference between mRNA and pre-mRNA actually may be quite significant for them. Okay? As we learn more about genetics, they learn more and more kind of specific things, but all the cool things that happen on the levels, right? And it's not just this to this to this. It's like all of these different cool things that happen. All right, so for 2b, what monomers? Monomers are the individual component. Mono is one. And so monomer is something that makes up a polymer. What are the monomers here? Yeah, those are called RNA nucleotides. Okay? So this is made up of RNA nucleotides. Nucleotides, you've seen that word before. You also have nucleotides in your, not just your RNA, but also your DNA, right? And we know that they pair, which we'll talk about in a second. For 3, according to Model 1, what enzyme is required for transcription? So I have an enzyme, and enzymes all end in what letters? ACE, yeah, they all end in ASE, so what's my enzyme here? Can you see an enzyme? Yeah, RNA polymerase, ACE, right? So my enzyme for number 3 is RNA polymerase. So really, for my central dogma, I should have kind of like a, I'm going to kind of make a little bit of a chart here of some of the key characters as we kind of as like an aside. So I actually know in order to make this happen, I need RNA polymerase. Okay. So, a simple name to remember though. ACE, it's an enzyme that makes RNA. Polymer is a long chain of something. So it's basically the enzyme that makes a polymer out of RNA. Okay. All right, number 4. Refer to model line. Okay. So A, what is the base pair rule for the DNA strand matching the RNA strand? So there's a base pair rule. And you may know this off the top of your head. If you don't, it's okay, but we're going to look at it here. So if my DNA is an A, so this is my DNA, my RNA will be A. This is the tough one. I shouldn't have started with this one. If I have A, it's going to be a U.
Well, if
It's a
RNA,
T if it's DNA. Yeah, exactly. But if it's RNA, it's going to be a U.
Yeah.
Okay? And if it is a T, the RNA will be a. So that the RNA, so if it's my DNA is a T, the RNA will be an A. Yeah. Okay? And if it's a C, my RNA will be a G. And if it's a G, my RNA will be a C. Okay? That's the base pairing rules. Okay. So compare this base pairing rule with that of DNA strand. So is it the same as DNA? There's only one difference. What's the difference? In DNA, so if this were DNA, it would be a T, right? The rest would be the same. But if this was DNA, I would have, my pair would be a T. All right, number 5. Which strand of DNA contains the blueprint for the pre-mRNA? So this actually involves us kind of understanding these different strands. See how one of my strands is a little dashed line? We're not really concerned about that. But I do have a strand. That's called my template strand. Okay? That's going to be my blueprint. And for 6, in which direction is the DNA molecule read? So this is a little bit confusing. And I got messed up the first time I filled this out. But I'm reading, am I going this direction or am I going that direction? I'm going that direction. So I'm actually going from 3' to 5', right? So these are the little ends of the DNA, and that's the length, that's the notation we use. I'm reading it from 3' to 5'. I'm reading it. That's how you would write it. That little thing means prime. Okay. And that's actually quite important in understanding when sometimes you get a big sequence of DNA in a lab, especially if you're working in a lab that has a lot of computer work with DNA. They're going to say 3' to 5' or 5' to 3', and you kind of just have to know, that just means the direction of DNA. Okay. So for B, the DNA strand and pre-mRNA strand are anti-parallel. With this in mind, label the 3' and 5' ends of the pre-mRNA. This was very confusing for me. Okay? This is the 3' end, and this is the 5' end. So what end of the RNA is this right here? Yeah?
It's the 3'. The 3'.
The 3' is correct because it's opposite that 5' end. So put a little 3' right here, and you can put a 5' right here. So that being said, in which direction is the pre-mRNA constructed? So which end was this? Because this was the oldest part. That's the Yeah, this is my 5' end, and the 3' is actually the newest part because this is still being made. So it's made in 5' to 3'. If you were to stop me on the street this morning and say, Mr. J, in which direction is pre-mRNA made? I couldn't tell you off the top of my head. I always need to look at a diagram for this, so don't think that you have to just know this. But if someone references 5' or 3' end, it's important for you to understand that they're just talking about the direction of DNA. Okay? All right, we're blowing right through this. Very impressed. And so for number 7, there's a key there. It must be important. Before printing presses were available, books had to be transcribed in order to share the information in them. Consider the definition of transcription and explain why the process in Model 1 is described using that word. So, transcription is to kind of make a copy of a book. What am I doing with my DNA? I'm copying it. Good, I'm copying it. I'm making a copy. Now that being said, I'm making kind of a cheaper copy of it. Because my DNA is like, that's the gold, right? That's the perfect stuff. I don't want to mess with that. My mRNA is basically just like a copy. And we're going to talk in a second why you need that copy. Okay? All right. So, for next, I have to read this on page 3. And this is, just bear with me, because we're going to get to a point where we really understand what an intron and an exon are. And that is stuff that I just think it's really important for us to understand, because I think in the labs that you work on, they're going to expect you know it. And I think we may have all heard of this phrase, but you may not totally understand it. So I want to just kind of get to there. So read this. In eukaryotes, the enzyme RNA polymerase and it joins with several transcription factor proteins at the promoter, which is a special sequence of base pairs in the DNA template strand that signals the beginning of a gene. The transcription factor proteins, along with the RNA polymerase, is called the transcription initiation complex. This moves along the DNA template strand about 40 base pairs per second, producing pre-mRNA. So this is going 40 base pairs per second. It's going super fast. When the RNA polymerase reaches the terminator sequence of the base pair and the DNA template strand, it completes the production of pre-mRNA and releases it into the nucleoplasm. So this is going to go until it reaches the terminator, and then it's going to be cut. Okay? So number 8, which part make up the transcription initiation complex? So I'm just going to kind of look here. My transcription initiation complex is the transcription factor proteins along with RNA polymerase is called the transcription initiation complex. So it's transcription factor proteins and RNA polymerase. Transcription factor. If you work in a lab that studies cancer, I promise you that they will call them TF proteins. And you will now know, it has something to do with making RNA. Number 9, where on the DNA strand does the transcription initiation complex form? Well, so where is it going to form? It's going to form at the promoter, right? What's a party promoter do? They're like, hey, come to my party, right? And that's what a DNA promoter does, right? They say, hey, come make this DNA right now, okay? Nearly all the cells in an organism contain identical DNA. The cells in my finger, do they have the same or different DNA than the cells in my eyeball? The exact same. Well, why don't I have a finger? Why don't I have an eyeball growing on my finger or a finger going out of my eyeball? Why is that different? Because what? They have gene regulation. Because I'm not actually reading the entire strand of my DNA. I'm reading the instructions for a finger in my finger, and I'm reading the instructions for an eye in my eye, right? So, is it likely that a cell would transcribe all of the genes within its nucleus simultaneously? Is my RNA polymerase going to read the entire strand of DNA? No way. Partially because those are on different chromosomes, right? But also because, am I ever going to need the information to make an eyeball on my finger? No, there are parts of that will never be used. Okay? And just as a quick note about that, I have some DNA that's kind of easy to be read and some DNA that's not easy to be read. and it's wrapped around these little dudes. You know what these little dudes are called? Anyone know? Yeah.
I think histones.
Histones, yeah. So these would be histones. And those are going to be quite difficult to read. So if my finger, the information for an eyeball is kind of wrapped around histones, meaning it's just packaged away in it's just in the attic, and it's never going to use that information. But the information to make a finger is right here. It's very helpful. It's accessible. So all of the cells in my body have the exact same DNA, but some of it's just kind of wrapped around different things. Okay, so for number 11, considering the many types of cells in multicellular organism and their different functions, is it likely that all cells transcribe all their genes at some point in their lifetime? Will my finger ever use the instructions to make an eyeball? No, right? They're only using the parts of their DNA that they need, okay? The rest of it's kind of just wound up. And now we're getting to a good place where we're kind of talking about mRNA processing. So I made my pre-mRNA, okay, which is this. And this is my pre-mRNA, but it's labeled with some nice little things. All right. So on this gets turned into just mRNA, which is what we kind of often hear. So this pre-mRNA, so my DNA, I'm going to kind of just do my little label here. My RNA polymerase will make pre-mRNA, and then that gets turned into mRNA, but some things happen to it. And we're going to try to figure out what happens to it. So for 12A, what has been removed from the pre-mRNA to make it into RNA?
Introns or non-proteins?
Yeah, so I just get rid of my introns, okay? For this, we'll just call them introns, okay? So I'll put a little minus sign. I lose my introns. This is something we used to call junk DNA, although that language is used less and less because it's not useless. Okay, 12B. What has been added to the mRNA that was not present? So here's my pre-mRNA. Something got added to it. Yeah.
The methyl cap at 5' and the polyadenine tail, which extends its length throughout.
Exactly. So I add in my methyl cap, and I add in something called my poly A tail. And this poly A tail is very important. When I'm about to go into my nucleus, I have these things called exonucleases, and they want to eat the DNA right away, or the mRNA right away. It's good that they do that, because they break it down, and so I can recycle everything. But here's the problem. If they kind of eat my RNA before it gets to the ribosome, then my protein's going to be all messed up, right? It's like I delivered a mail, and then there's these little kind of like dogs that are eating it, and they eat half my letter. I'm not going to understand that letter. So the kind of solution is, well, I'm going to bring all these little dog treats, right? And so those dogs can just eat those treats, and they're not going to actually damage my letter. Okay. So looking back at this, number 13, identify the structure through which the mRNA leaves the nucleus. It leaves through the nuclear pore. Number 14, the nucleotides and the mRNA will be read in the next step, producing a polypeptide. Which sequence of bases on the starting point indicates the starting point for the blueprint? We have that label there. It is AUG. And AUG up here is my start codon. All right. Now 15. The M in mRNA is short for messenger. Why is this called messenger RNA? It's just messaging the information of the DNA out to the ribosome. Okay? That's why we call it the messenger RNA. Beautiful. We got to the point where we needed to, where we're going to talk about introns and exons. We're kind of getting to a point where we're understanding at a deeper level what's happening with our DNA. So, my introns, this is on page 5, are sections of pre-mRNA that are non-coding. That is, they don't provide useful information for the production of the polypeptide being synthesized. That polypeptide is my protein. Okay? So they don't get made into a protein. There is evidence that suggests that these introns allow certain sections of DNA to code for different polypeptides when different sections are removed. So I want you to think about this for a second. See all these different introns I have? And these can kind of combine these different exons. But if I only remove some introns or different kind of combinations of them, it actually allows me to make these really cool variability of proteins. And we no longer think that these are just junk DNA. We think that we're starting to find out all these useful things. But if you think about it, they kind of break up my DNA into, instead of one long strand, all these different things that can make all these different cool combinations. The removal of specific sections is triggered by a signal response in the cell. The portions of the pre-mRNA that remain are called the exons. So this is all my exon. The methyl cap, sometimes called the GTP cap. Oh, this is the GTP that you were referencing. Helps the mRNA molecule move through the nuclear pore and attach to a ribosome. Its final destination. mRNA is a short-lived molecule. Once in the cytoplasm, the mRNA will be subject to exonucleases, like I was talking about, that immediately start removing individual nucleotides from the 3' end of a nucleic acid. The individual mRNA nucleotides will then be free to be used again during the process of transcription. Okay? So I'm going to stop us there, just for the sake of time. We're going to move on to the gene expression. Translation. You've seen this chart before, probably. Let's jump into this, okay? What do the letters U, C, A, and G in Model 1 represent? Those are, yeah, and what kind of nucleotide? Yeah, mRNA nucleotides. What do the abbreviations such as PAG, ILE, ALA, and GLY, all of these abbreviations stand for? Amino acids. The language of mRNA is often described as a triplet code. Explain the significance of this reference. If I have triplets, how many kids do I have? 3. And these are made up of 3 letters. So it's triplet code because it's 3 letters. All right. For number 2. If an mRNA molecule had 300 nucleotides in the coding region of the strand, how many amino acids would be in the polypeptide? Polypeptide is a protein that was synthesized. If I have 300, how many proteins can I, or how many amino acids can I make? Well, if 3 makes 1 amino acid, how many amino acids would I make? 100. All right. For number 3, let's look at Model 1. How many different codons code for the amino acid proline? So let me find proline, which is right here. And I see that CCU, CCC, CCA, and CCG all make protein. So 4 different codons code for proline. Let's compare all of the different triplets. Well, the similarity is that they all start with CC, and the difference is that they all end in a different letter. Sometimes mistakes happen here. Why do you think it's good for us to have 4 different letters that, or sorry, 4 different triplets make the same thing, considering that mistakes happen? Yeah.
To minimize the number of mistakes that actually have an effect.
Yeah. Let's say I'm gonna give you some directions on how to make amino acid. You're gonna do C is the first one, and then C, and the next one is. And you didn't hear it, right? But could you make the right amino acid? Even though that mistake happens? Yeah, you would make the same amino acid. So you would have, your protein would still be the same protein. That's nice, right? This is a big complicated thing. It reads super quickly and a lot of mistakes happen. So the fact that if you get the first 2 right, you're good for some of these, that's a nice thing, right? All right, using the mRNA codon chart in Model 1, we're gonna complete the following. So, I need to make my protein now. So I have my mRNA, and then this is going to get constructed. And so I have my TAC, and that what RNA is that going to correspond to using our rules? T pairs with? A pairs with?
T.
That's for DNA, but for RNA it is U. And C pairs with? G. Okay. And then next, I could talk about the polypeptide sequence that responds to AUG. So here's how you read this. A, first one. Second one is U. And then G, which is MET, or the start codon. Okay? So that is MET. All right, the next one is CTT. C corresponds with? G. A, or T is? A. And now GAA is? A is GLU. Okay, next is CGG. So looking at CGG. CGG, don't worry, I'm not going to do all these. Is ARG.
You didn't.
Oh wait, sorry. I didn't. What was my mistake? I made a mistake. Yeah, I didn't transcribe it. I'm sorry, everybody. Now GCC. Okay, I'm getting tired of this. GCC is ALA. Okay. Now, I'm not. For the purpose of this, if you kind of understand that, that's really all I need you to get to. But let's look at the very last one. The last one is ACT. That corresponds to UGA. And looking at UGA, I have UGA. Ah, that confused me. Stop, right? So that's kind of important because that tells you to stop. It's the terminator, right? It's the kind of things that ends it. It's not actually the terminator in this example. Okay, so looking at this, what is the amino acid at the beginning of every polypeptide for question 5? It is MET. Okay. And you don't have to write down an answer for 6, but almost all living species follow these rules. And scientists use that as evidence that we probably came from a common ancestor, right? Because we all follow, all of the very, very different things on Earth follow these very, very different, or very, very, very similar rules. And so we use that as an example that we all kind of come from common ancestors. Okay, beautiful.